Okay, so today we're going to take up all about the different method for inoculating your organisms. The first one, we have here your streak method here with your clock streaking method. Okay, procedure for that, you'll be needing here. Okay, so we have your bacteria inoculum. This one is placed in your screw cap container. So, pwede din siya galing sa ating plate kung doon na nakalagay ang ating bacteria inoculum. But in the case where you have here your bacteria inoculum placed in your screw cap container, okay, another thing you'll be needing here would be your flame. So you could have here your Bunsen burner, you could also have your alcohol lamp, or you could have your others would have commercial na mga flaming na mga devices. But you can use here your Bunsen burner. You can also use your alcohol lamp for that. Another one, you'll be needing also here your loop or your needle. Inoculating loop or your needle. So pag sinabing inoculating loop, so expect na dito sa dulo niya may ganyan, may butas. Pag needle lang siya, so needle lang siya, ganun. Pointed, that's your needle. But if you have that one na bilog sa dulo, so that's your inoculating loop. Okay, so okay, the first thing you're going to do here is to properly hold your inoculating needle or your loop. So, the proper way of holding that one, so parang ganyan, parang sumusulat lang tayo. Again, hindi pwedeng ganyan ha. Hindi pwedeng ganito ang pag-hold, but rather it should be, okay, you hold that one in such a way na parang ganyan, parang nagsusulat tayo. Ganyan. Okay? Okay, then you're going first to flame your inoculating loop. You hold that one, so ganun nan. And try to flame this one by allowing to pass through pag ganun-ganun mo siya. Okay, allowing here the end portion here to be flamed, okay, by your Bunsen burner or by your alcohol lamp. Allowing it to pass through several times pag ganun. Okay, then next thing here, you're going to get here sample from your bacteria inoculum in your screw cap container. So the proper way of getting a sample for that, so like for example, since wala ako dito na screw cap container, for example, itong screw cap container ko. Okay, so if the one holding your inoculating needle here is going to hold, okay, so ito gagamitin mo your little finger in the hand holding your inoculating needle. You're going the one to open this one. So pag ganun, ayan. Okay. So hindi pwede na ganyan pag-open mo lang. Okay, kahit ano na lang. No. Again, if this one holding your inoculating needle or loop, so itong little finger mo, ang index finger mo ang going to, okay, so paikot mo lang ito. Okay, then, ganyan. Then, okay, so before you get a sample, if this one contains your bacteria inoculum, before you get a sample of that, i-flame mo muna ito. Okay, so papadaanin mo sa flame. Okay, papadaanin mo sa flame ang mouth nito. Then you get a loop full of your sample on that. Kukuha ka ng sample on that. Okay, i-flame mo itong mouth nito. Okay, i-flame mo ulit. Okay, with your Bunsen burner or your alcohol lamp. And then try to cover. Pabalik. Okay, so ganun. If you have already your, okay, if this one already nakakuha ka na ng bacteria inoculum on that, the next thing you're going to do here is try to, okay, to put this one, you're going to streak now on your petri dish or your agar plate containing your agar. So, for example, ito ang plate natin. So, you're going to remove the cover. Okay, so slightly remove that one. Okay, tanggalin mo ang cover nito. Okay, try to remove the cover. So, kung may cover ito, for example, ganun. Slightly remove that one. Okay, so wag mong masyadong lakihan ng pag-open yan to prevent that one to be exposed to the air because remember the air here contains your contains the microorganisms. So, medyo open mo siya, lightly lang na open. And then try to flame the mouth of this one. So ipapadaanin mo ulit ito sa ating Bunsen burner flame. Okay, itong area na to. Then after that one, since you have already your inoculating needle na may bacteria inoculum, so you're going to start here with your clock streaking na method. So we start here dito sa isang edge. You can divide actually pwede mo itong guhitan dito sa gitna ang ating agar. So pwede mo dito sa likod niyan or sa baba niyan, lagyan mo ng ganyan na drawing. So we start with the first quadrant. Okay, the first quadrant. So dito ka muna. Ganyan yung start dito. Ganyan na streak. And then ganyan. Okay, that's your first quadrant. After you streak that already, you try to cover. Cover mo ulit ito. Before you cover, i-close mo ito, i-try to flame mo ulit ang mouth nito. Okay, ulit, pag nag-streak ka na, i-flame ang mouth nito ng petri dish natin. Try to cover. Okay, and then your inoculating loop, okay, i-flame mo din siya. Or pa ganon. Allow this one to cool. Okay, next thing here, you're going to streak on the next quadrant. Okay, open mo ulit. Slightly open lang. Then after that one, pag na-open mo na, flame the mouth first of your, of your petri dish. With your inoculating needle, flame mo siya ulit. Okay, kukul mo muna siya bago ka mag-streak ulit. Okay, so we are going to cool that one para hindi mamatay ang bacteria natin. Okay, then, pag na-cool na siya, ang flame mo, ang inoculating needle mo, or your loop here, you try to start the second quadrant streaking. So, kukuha ka dito. So, i-rotate mo na ngayon ang ating petri dish. Okay, ganyan. Then, kukuha ka ng sample dito. Small lang. Dito na part ha. Kukuha ka. Okay, then, try to streak this one pag ganon. Okay, then after that one, try to flame the mouth of your petri dish. Tapos, close this one. Okay, with your inoculating loop or needle, if you flame ulit, then try allow to cool. The next one, you are going to, ready ka na ulit na mag-streak ulit. You turn again your petri dish into the next quadrant. Okay, slightly open. Flame the mouth. Okay, and then flame your inoculating needle or loop. Allow it to cool. Okay, and then kukuha ka ulit dito ng part of your bacteria. Okay, kukuha ka ng konti dito, then try to streak. Wait lang. Ito pala. Kukuha ka dito ng konting bacteria. Okay, so pag ganun siya. And then, streak mo ulit ito. Okay, then next, ulit-ulitin mo lang until you have already. Ganyan. So, make sure here, ang ika-last na quadrant, so hanggang dito ka lang, huwag mo siyang i-overlap dun sa kabila natin. Okay, then, if you are already finished with all your streaking on the different quadrants, okay, try to cover this one. Okay, and then, before you close that one, you try to flame the mouth. Okay, then try to cover. Okay, then with your inoculating needle naman, if i-flame mo din siya. Okay, and then allow it to cool. Okay, and then itatago mo na siya. So that would allows you to basically kill the bacteria and to sterilize your loop. Okay, then after that one, you are ready now to incubate. Pag nag-incubate tayo ng plate, okay, you try to put that one in upside down. Okay, so mintos ibabalik ta rin mo. Mintos ang agar mo ay nasa taas. Okay, and then try to put that one in the incubator. Hindi pwede na ang agar mo sa baba. Mintos ay ibabalik ta rin mo ang petri dish na may agar natin which has already inoculated na organism and try to put that one in your incubator. Okay, incubate that one to their corresponding na temperature, approximately 35. Pwede siyang 37, but most likely we are setting incubator 35 degrees Celsius. And try to incubate on their corresponding na time duration, usually 24 hours incubation natin. Okay, the next one we have here, inoculating your organism in your broth na culture medium. Okay, For this one, so just need first, again, yung flame mo ang ating inoculating loop or needle. Okay, kukuha ka dito ng bacteria inoculum. Paano ulit kumuha ng bacteria inoculum? So, ganon siya. Okay, holding this one with little finger. Okay, open this one by ganyan. Okay, and then okay, ano mo muna siya, i-flame ang mouth nito, dito na part. Kukuha ka ng bacteria inoculum in that. Okay, before you close this one, i-flame mo ang mouth. Close mo ito. Ganon. Okay, if you have already your inoculated organism in your inoculating loop or needle, so pag broad medium lang siya, iganon mo lang siya. Insert this one approximately 1/4. Okay, so 1/4. So pag 1/4, dito lang siya, mga dito na part. So, ganun lang siya. Ipagano mo dito. Okay, then you can rotate this one, ang inoculating loop natin dito. Okay, rotate mo siya dito. That will allow your bacteria inoculum na dito sa inoculating loop here to be present in your broth na medium. Then after that one, okay, you can flame them out of this one. If this one's screw cap container. Okay, and try to cover. Okay, and then flame this one. And then try to incubate them. Incubation, so same incubation. 35 degrees Celsius here for a specified time duration depending on your bacteria. Okay, how are you going to inoculate here in your bath na culture medium? So again, with your bacteria inoculum, with your inoculating loop, i-flame mo yung ating inoculating loop. Okay, then kukuha ka ng bacteria inoculum dito. Okay, so again, every time you open, you flame. Okay, so after closing, you flame. So, ganun din dito sa mga culture media natin. Before you open that one, okay, you can flame. You open, you flame. Okay, then you close, then you flame. So, ganun ang ating aseptic technique. Again, that's to prevent the entry of your mga contaminants or those organisms from the outside environment. For the culture media, so ganun din siya. Okay, so with your inoculating loop, okay, try to stop here. Yung stop natin up to 1/2. Okay, so stop mo dito lang inoculating needle natin, then tatanggalin mo lang siya. Make sure na you will not able to touch ang mga side ng tube natin. Okay, then after you try to inoculate, flame the mouth, cover. Okay, and then inoculating needle na ginamit natin, flame natin. Okay, para mamatay ang bacteria natin. Then try to incubate on their respective na temperature and their time duration. Okay, for the regular slant, so this is your regular slant. So procedure, first you flame your inoculating needle. You get bacteria inoculum in your screw cup container. Okay, so again, open the screw cup, flame the mouth, kuha ng bacteria inoculum. After pagkuha, okay, try to close. Okay, and then flame the mouth of that. Okay, and then try also to, kuha kaya, then try to flame the mouth and try to cover. Wait lang. Okay, open mo ang screw cap container. Bago ka kumuha, i-flame mo ang mouth. Kukuha ka ng bacteria inoculum. Before mo siya i-close, i-flame mo muna bago siya i-close. So, same with your mga culture media natin or mga plates. Pag nag-open ka ng plate, bago ka ma-inoculate, okay, open the plate, or this one, open mo din, flame. Okay, then, inoculate. Okay, then, before you close, flame. Ganun palagi ang technique natin. Okay, for the regular slant, how are you going to streak or how are you going to inoculate your organism here? So, you start at the bottom portion. Ganun lang siya. So, you will just touch here the surface. Make sure na hindi mo pinapuncture ang agar. But rather, in the surface ka lang. So, kung ito ang surface mo, so dito ka lang magta-touch. Okay, so kung dito ang agar mo sa dito, huwag ka dito mag-puncture, ha? Okay, so dito ka lang magta-touch dito sa surface na to. So, start with the bottom. For example, dito ang bottom natin. Okay, start with the bottom here and then you just need to draw a straight line lang. Pag ganun. Okay, so pag ganun lang siya. Other technique, pwede siyang fish tail. Pag ganun. But I think ideal ang ating straight line lang. You draw a straight line on the surface. Ganun lang siya. In the case of your special slant, how are you going to inoculate? Again, remember the special slant, meron siyang butt area and meron siyang slant na area. So for this one, first you need to stab here on the butt area. Okay, so 1/2 lang ito ulit. Pag nasa butt tayo, always 1/2. Okay, ng ating inoculating needle here or loop. Inoculating needle, we're able to puncture the butt gelatin. The agar, the agar in the butt region or area. Then you take this one, tanggalin mo, and then continue on the surface of the slant. You draw a straight line dito pag ganon. So, pag makakita tayo ng colony growth, so makikita tayo ng colony growth dito, plus makikita ka dapat ng may colony growth ng pagganyan din. Again, you stop on this area, tatanggalin, then try to continue on the straight line on the surface of the slant area. Okay, then, flame your loop, and then try to incubate corresponding the temperature at time durations specific for each of your bacteria. Okay, so that's your inoculation of the organism here in our different culture media. Okay, thank you.